So I will start my class today, and in this class I will continue from where I had left off in the last class. So with this I will present my screen. So the place where I had left off, I was modulating access to genes. We know that from the last class we knew that gene expression is differentially regulated. So different cells have got different genes which are expressed. Therefore, even though they have the same set of genes present, but they express different proteins, because not all genes are expressed similarly in different cells. So one thing which we know, that the genes are located on DNA, the DNA, and the DNA are on chromosomes. So, and you know the chromosomes are tightly packed materials. So how do transcription factors find access to these genes? This is a question which we need to answer. How do transcription factors find access to genes? That is, to the promoter of the genes. How do they find access? So that is what we are talking about, modulating access to genes. So there is a difference between prokaryotes and eukaryotes. Remember that this class also is covering the topic in molecular biology, eukaryotic gene expression, as well as differential gene expression. So in prokaryotes you have inside the cell the DNA arranged mostly circular, like this, and very little nucleosomal or compaction is present. There are some nuclear proteins which are present, but very little. So these DNA is more or less always accessible to the transcription factors. One more thing is that transcription and translation are closely coupled. That is, where from this DNA, this is the DNA strand, this mRNA is being made over here. This mRNA is being made, and while this is made, ribosomes bind over here and they make the proteins. So transcription is occurring, and while the transcription, the RNA tail comes out, the ribosomes bind and start to make proteins. So the transcription and translation are tightly coupled in prokaryotes. In eukaryotes, the situation is different. In eukaryotes, you know that all the genes are arranged on chromosomes. They're on DNA and they are arranged in chromosomes. You have read a lot about chromosomes. So in chromosomes, how do transcription factors find access? In eukaryotic, in eukaryotes, eukaryotic genes are contained within a complex of DNA and protein called chromatin. So a complex of DNA and protein together they form the chromatin. and this protein component is predominantly made up of proteins known as histones. They are made up of proteins known as histones. And then we have a structure called the nucleosome, which is the basic unit of chromatin structure. And the nucleosome is made up of an octamer of histones. So histones can be H2A, H2B, H3 and H4. So the main nucleosome consists of this histone octamer. So these 8 histones together, they form the 8 histones together, they form this nucleosome core. And on this, the DNA is wound. And the DNA is wound with 2 loops containing approximately 147 base pairs of DNA. Histone H1 is bound to the 60 to 80 or so base pair linker DNA between the nucleosome. So you have, this is one nucleosome, another nucleosome, histone core. You have the DNA coming. Let's see if I can use another color. You have the DNA coming over here. After roughly two turns, the DNA comes over here. And you have histone you have the histone H1 binding in this region. This region in between the two nucleosomes is known as the linker. It is known as the linker. Let's have a look at the figure. The color needs to be changed back to red. Let's let's make it a light color here. So you can see this gray region here. Around there are 2 turns. This is the DNA. And you have the histone octamer H2A, H2B, H3 and H4 present 2 times. H2A is present, 1 on the top, 1 bottom. H4, 1 on the top, 1 bottom. So you have basically an octamer. So 2 into, you have H2A, 2 into H2B, H3 and H4. So this makes this deep green octamer over here. And you of course have the H2A tail, H2B tail, H4 tail, H3, because the protein is, say this is a histone, it's folded into this structure, and you have this region is the C terminal region is the tail, and the N terminal region is the head of the protein. So you have the tails of all the octamers. Now you find these nucleosomes, they are attached like this, and the DNA is wound here, it's wound here, wound here, wound here. They can take a structure like this, which is a solenoid. This is a solenoid structure. This is a solenoid structure takes place, and this solenoid structure is organized like this. This is a solenoid, and solenoid, another solenoid, another solenoid, another solenoid. So they are organized like this. So this is how the basic packing takes place. And you can see these are the nucleosome, the nucleosomes. This region in between, this is the linker region. This is the linker region. And on this linker region, this orange colored are the H1 histones. So the H1 histones bind in this region, in this linker region, and they bring all the nucleosomes in a more compact manner. Sometimes you find these tails. The tails of these histones are methylated when the nucleosome is condensed. So methylation causes condensation. It gets condensed like this. If the tails are acetylated, that is CH3CO group are there, then they are uncondensed. They are uncondensed. So if they are very much condensed, then histones are bound and very much condensed. So where will RNA polymerase bind and do transcription? It won't be able to do transcription. And no transcription, no translation, no protein. Gene is not expressed. So RNA polymerase won't be able to transcribe. So if you methylate a particular gene, that region of the gene, it becomes condensed in the nucleosome, and it will not be expressed. But if you acetylate these tails which are coming out, then they are uncondensed, and then the DNA is free, and then RNA polymerase can bind over here, but then this also unwinds later. So histone tail modifications, they control in condensing and uncondensing and making genes either accessible or unaccessible for the transcriptional machinery to bind. So this is a picture of histone methylation on histone H3. So you must practice this diagram. So there are some regions on the histone 3 tails where methylation, when it takes place, which is important for one is for one region methylated cell cycle regulation, one is required for transcription elongation, one is required for transcriptional memory, then there is a silent heterochromatin, then transcriptional activation. So all these tails have got regions which are important, and basically these histones are rich in lysine and methane. They are basic proteins because nucleic acids, they are acids and bases, they bind. Next, so there is a memory of methylation. So the modification of histones can also signal the recruitment of proteins that retain the memory of the transcriptional state from generation to generation as cells grow through mitosis. So sometimes these methylation, there is a memory of methylation. There are methylation rich spots. So when the cell divides, this methylation is also carried forward to the next cell. So the next cell, when one cell divides into two, this memory that this was methylated over here, it is carried to the next cell. Examples are the important proteins known as trithorax and polygom family proteins. When bound to the nucleosome of active genes, trithorax proteins keep these genes active, whereas polygom proteins, which bind to condensed nucleosomes, keep the genes in a repressed state. So polygom proteins repressed and trithorax proteins uncondenses and active, gene is active. So the polygom protein falls into two categories that sequentially act in repression. The first set has histone methyl transferase activities that methylate lysine in histone H3. Therefore H3 lysine's formula is K. So lysine, the 27th lysine of histone 3 is methylated, and the 9th lysine is also methylated, and they repressed when these two lysines are methylated on the histone 3 tail. When these lysines are methylated on the histone 3 tail, on the H3 tail, this is the H3 tail. On this H3 tail, where this H3 tail, if these two lysines, that is K9 and K27, when they are methylated, and their methylation is by the polygom family of proteins, and when they are binding over there, they methylate it and the gene is condensed and repressed. This is how it works. This is one form of whether it is methylated or non-methylated. In some cells, is maybe in muscle cells, maybe these polycoms, they methylate this region. Maybe in liver cells, they don't methylate these regions. So muscle, in muscle, where the methylation has taken place, it becomes inactive. These genes are not expressed, and in liver, methylation has not taken place, it is active. So immediately you can see a differential gene expression taking place. So now let us go to the anatomy of a gene, eukaryotic gene specifically. So let us talk about the beta globin gene. So you know there are exons and introns present. You have a region before transcription takes place. This region is known as the promoter region. This is not transcribed. This is a regulatory region where TBP site RNA polymerase binds. RNA polymerase binds to this region. There are also other sites where other transcription factors bind and RNA polymerase bind. The Tata binding protein site with, where all the this TBP, where all the RNA polymerase binds over here. Then there is a transcription start site or transcription initiation site. Immediately after, it is not the translation initiation site. And you have a leader sequence also known as untranslated region, and then the ATG or AUG starts, where the proteins start from here. The methionine and other proteins, they start from this region. This is the coding region. Apart from that, this region which is which is transcribed is but and it and also later it remains. It is known as exons, and there are also regions in between these exons known as introns, which are spliced out. They are removed after the transcription. So and then in the end you have a poly A addition site. You have a translation terminator codon, or TAA, which is also present here, and transcription terminates here. This is transcription start, transcription end. In bacteria, if you can remember the row and row independent, in eukaryotes you also have a similar mechanism present. Then you have the nuclear RNA, which comes out immediately. Already splicing has not taken place, just transcription. The exon 1 has been transcribed. Also it has, this is the translation start site. It is not, we are not talking about translation. And intron 1, then exon 2, intron 2, exon 3, and you have the poly A tail in the end. A polyadenylation signal, poly A tail. Processing takes place and the introns are removed. And then ribosome binds and you have the translation taking place. Then you have post-translational modification, methylation, acetylation, sumolylation, different types of things take place, and this beta globin, it folds into its secondary and tertiary structure, and it binds with alpha globin and another molecule, and you have the eventual hemoglobin protein formed. This is the best example of an eukaryotic gene which you can study. This note you can even transform, you can even apply in your eukaryotic gene in your molecular biology paper. I am mentioning this time and again. So this is the anatomy of the gene. All these things you have to read. In the next class, I will go ahead with cis regulatory elements, where the on, off and dimer switches of a gene, they work. Please, I would request you to please keep on studying, or at least watch my videos time and again, so that you can at least read all these things. And as you know, you can get this from LibGen site. You can get Gilbert's book, and you can read them over here. Chapter number 3 in Gilbert's book. You can absolutely, you can download them from libgen.is site. You can type Gilbert develop and download the PDF and read from there. I will pass my notes on later, but keep on reading, and I will start the next class on the next day according to the schedule. Maybe I won't be able to take in the morning because I have a lot of work in the morning, but so I'll take it in the afternoon and evenings. Do you people have any questions? আর কোনো কোয়েশ্চেন আছে? Aditri, any questions?
No, sir.
Okay. Then I'm ending my class over here. Wishing you all the best. I will send you the meeting link.